amino acid in the chain, the only way we can do that is by taking this polypeptide chain and then adding it to this right here. We can't take this and add it to this end because this was the very first amino acid in the chain. And so we need this to be behind this circle guy right here. So the only way we can do that is by taking this polypeptide chain and transferring it to the tRNA molecule in the A site. Once we transfer that polypeptide to the amino acid in the A site, that's when this shift is going to occur. So we said that the P site is responsible for holding the growing polypeptide chain. Well, it is up until this part. So there's going to be just a little bit of time where the polypeptide chain is going to be in the A site. But then once the ribosome shifts, the tRNA stay put and the mRNA stays put. And so that's going to ensure that what was in the A site is now going to end up in the P site. And that empty amino acid that was in the P site, once you shift, now it's going to be in the E site, in the exit site. So now it's going to be able to leave, go get hooked up with another amino acid, and then, can come back and contribute to making more protein. And then the cycle repeats. All right, so we grow in polypeptide chain, we bring in a new amino acid, we have peptide bond formation, so we're going to transfer this polypeptide chain from the P site, and we're going to transfer it to the tRNA in the A site. And so that A site is temporarily going to hold that growing polypeptide chain. After peptide bond formation, that's when we have this translocation, or this shift of the ribosome over. And that shift is what puts the proper tRNAs in their proper place. So after the shift, now you've got the A site empty for the next amino acid to come in. You've got the P site holding your growing polypeptide chain. And then now that empty tRNA molecule that just donated the amino acid is now in the E site, so it can leave. Does this kind of make a little bit of sense? All right. Like I said, this is the animation that I'm going to show you because this is definitely kind of the trickiest to visualize. Lots of moving pieces and lots of moving parts. Okay. And then finally, termination. That's going to be the last stage of translation. And remember, termination is going to be dictated by these stop codons. And so you are responsible for knowing your start and your stop codons. So in case you've forgotten what they are, the stop codons are UAA, UGA, and UAG. So anytime a ribosome comes across one of these stop codons, that's going to initiate this termination process. And in this termination process, what's going to happen, the ribosome, it'll recognize the codon, it'll recognize it as being a stop codon. And since the stop codons, they don't code for any amino acids. But instead, once the ribosome sees a stop codon, instead of signaling to a tRNA molecule, it'll signal to a different kind of protein called a release factor. And the job of that release factor, it's actually going to add a molecule of water to this growing polypeptide chain. It's going to hydrolyze or break apart that polypeptide chain from its tRNA molecule. So the release factor is releasing the protein from the tRNA molecule that it was attached to. And then after that, after the protein's released, then then translation is done. Then everything kind of dissociates. The small subunit comes off, the large subunit comes off, the mRNA is released until you transcribe a new mRNA and you need to make a new protein. All right, questions? Got anything?
So you said the release factor add a molecule of water?
Yeah, the way that it releases this polypeptide from this tRNA is by hydrolysis. So it literally adds a molecule of water, because remember, the dehydration synthesis and all that stuff, or hydrolysis, you add a molecule of water, and that's going to break this bond between the growing polypeptide chain and the tRNA molecule. So stop codons bring in a release factor that help release the polypeptide from the tRNA molecule. Yeah, Mark?
So when does it bring the water?
when does it bring in the water? That's what this guy does. That's what the release factor does. So the water, I mean, there's just kind of water everywhere. But he's specifically responsible for helping remove that polypeptide chain from the tRNA molecule. And then once that happens, then everything else just kind of dissociates and kind of falls apart. Other questions? All right. So, that was translation. Now, we need to talk about some other topics that are associated with translation. So, for instance, you know, when we're talking about gene expression, you know, that involves transcription of a gene and then translation of that mRNA. But what if we need to make multiple copies of a protein, but we don't want to waste a lot of time doing it? So instead of our, you know, our ribosomes going along, making a protein, starting over, making another copy, we can have multiple ribosomes attached to the same mRNA. So you can have these little trains of ribosomes, each making a copy of the protein. So if your cell needs 20 copies of, I don't know, protein X, then you're going to have multiple ribosomes that are going to be able to make those multiple copies. So imagine, so here's the 5' end of the mRNA, and imagine this guy was the first guy to come in and start translating. Well, he starts here, and he starts making his polypeptide chain in purple here. So once he gets started, then that leaves room for another ribosome to come in and start doing the same thing. So this was the second guy. And then once that gets a little bit further, then you bring in another one. And then once that gets a little bit further, you can bring in another one. So you end up with multiple ribosomes attached to an mRNA. And so this is going to be a very efficient process for making multiple copies of a protein. And this whole assembly line right here, this is called a polyribosome. Poly means many, ribosome is ribosome, so it's showing multiple ribosomes attached to a single mRNA. And so you can actually see this in this picture right here. All these little kind of, you know, blobs, these are all the ribosomes. And then the mRNA is the spaces in between the ribosomes. And so this can happen in prokaryotes and in eukaryotes, because all this is happening outside of the nucleus. So you can, at any given time, you can have multiple ribosomes on one piece of mRNA making multiple copies of a protein. Now, in prokaryotic cells, they don't have a nucleus, right? And they don't have RNA processing. So that's the difference between prokaryotes and eukaryotes. And because of those differences, bacteria, they not only can have these polyribosomes, but they're actually able to link transcription and translation so that they're occurring simultaneously. Because in eukaryotes, you know, we have to process the mRNA and ship it out of the nucleus, so there's no way that transcription and translation can be occurring at the same time. But in prokaryotes, because they don't have a nucleus and everything's kind of all happening in the cytoplasm, you can get this concurrent transcription and translation. So what will happen, you know, we have our DNA, we have our RNA polymerase, which is helping to transcribe the DNA. So here's our piece of DNA. The red pieces, that's going to be our new mRNA. And so as these RNA polymerases are going down, they're making this new mRNA molecule. But before the mRNA molecule even gets finished being made, all of these ribosomes are going to jump on and start translating. So you get this coupling between transcription and translation in bacteria. But the only reason why this is possible is because the bacteria don't have a nucleus and because everything's all occurring in the cytosol or in the cytoplasm. And so eukaryotes are not able to do this. They can have the polyribosomes, but they can't have simultaneous transcription and translation occurring. And so here you can see this again. You can see the DNA molecule very lightly. You can see, and then now you can see all these polyribosomes with the mRNA, you know, holding these ribosomes, or I guess the ribosomes attached to the mRNA. And so again, you get multiple copies, not only of the protein, but now you're getting multiple copies of the mRNA. So this is even more efficient. Because in eukaryotes, we still have to, transcribe one gene, then process it, then ship it out of the nucleus, and then we can make multiple copies. But in bacteria, you can make multiple copies of the mRNA at the same time, unlike in eukaryotic cells. Yeah?
Can you say how the polyribosome work again?
How the polyribosome works? Yeah, let's go back a slide and look. So we've already made our mRNA, right? It's already been processed in eukaryotic cells. And so when it goes to meet up with the ribosome, who's to say that only one ribosome can attach to one mRNA at any given time? And so we have a ribosome will come in, it'll start translating, start making this, protein in purple. And then as it moves down the mRNA, it's going to leave room for another ribosome to come in and bind to the mRNA and start making another copy of that protein. And then another one will come in. And so it's just like a little factory, little assembly line. And each ribosome is making its own copy of the protein. So this is going to occur whenever you need to have multiple copies of one protein. Now, if you only need one protein, then there's no point of having the polyribosome, right? You only need one ribosome. But a lot of the times, your cell needs to all of a sudden make a whole bunch of something. And so it'll use this polyribosome to do that. And so this is going to happen in prokaryotes and eukaryotes. But when it's coupled with transcription, that's only going to occur in prokaryotic cells because everything's already occurring right next to one another. Unlike eukaryotes where you have to process the RNA and then ship it out of the nucleus. Does that make sense? Okay. Any other questions? All right. Okay, so the next thing we're going to talk about are mutations. And we've kind of, brought this up over the course of the semester, but now we're going to get into the various different types of mutations. And so a mutation, all it is, it's one nucleotide getting changed into something that it's not supposed to be. So that's a mutation. And there are, the type of mutations that we're going to be talking about are called point mutations. And they're called point mutations because they're only mutated at one point or at one nucleotide. And so of the two types of point mutations, we have substitutions. That's where you substitute one base pair for another. In other words, if I'm supposed to have a T there and it gets mutated to form a C, that would be an example of a substitution. You're substituting the wrong nucleotide for the correct one. The other type of point mutation is called an insertion or a deletion. So this is where you're actually adding an extra nucleotide that's not supposed to be there, or removing a nucleotide that is supposed to be there. And so these insertions and these deletions, these often lead to frame shift mutations. Remember we talked about the reading frame and how it determines which amino acid is going to be produced. And so when those amino acids, those codons are being read by the ribosome. So if you insert an extra amino acid, you're going to throw off the reading frame. And if you throw off the reading frame, then you're going to end up getting all the wrong amino acids into place. And so we're going to go through, we're going to look at examples of these various different types of mutations. All right, so the two types of mutations are grouped into substitutions and insertions and deletions. So the first ones we're going to talk about are these substitutions. And again, a substitution is just when you replace a nucleotide with the incorrect one. Now the first mutation that we're going to talk about is a silent mutation. Now a silent mutation is going to change the DNA at one nucleotide. And even though you're having a mutation and you're changing a nucleotide, due to the redundancy of the genetic code, you're changing a nucleotide, but it's still coding for the same amino acid. So in other words, it's going to be in that wobble base that there might be a mutation, in that third nucleotide of the codon. And so if you have a mutation in there, but all four different possibilities code for the same amino acid, you're still going to end up with the same amino acid. Yes, it's still a mutation, but you're not changing the amino acid. So that's why we call it a silent mutation. It is a mutation, but it's not messing up anything. So no harm, no foul. Now, when you do end up changing the amino acid sequence, that's going to be a missense mutation. And then it's also possible to make an early stop codon. So if you have a mutation that leads to an early stop codon, then that means your protein is going to be cut short, right? Your protein's cut short, it's not going to be very functional, now is it? And then of the insertions and the deletions, again, these are going to lead to these frame shift mutations. And these frame shift mutations are so detrimental because once you mess up the reading frame, every single amino acid after that insertion or deletion is going to be messed up. And so that's really going to jack up your protein, which means it's not going to be able to work like it should. And so we'll go through and we'll look at examples of all of these. So at the top, we're always going to have the wild type. The wild type is like normal. This is what normally exists in nature. So we're looking at this particular piece of DNA. All right, we've got methionine, lysine, phenylalanine, glycine, and then a stop codon. So in this case, we're going to be looking at a silent mutation. So let's just say we have this CCG right here. And we mutate that third base in the codon. So we mutate it to an A instead of a G. So now this codon is going to be CCA. And so when we transcribe that, it's going to be GGU and instead of GGC. But GGC and GGU both code for glycine. So you're still having a mutation, but you're not affecting the overall amino acid sequence. So that's a silent mutation. Now, the missense mutation, again, we have our same wild-type piece of DNA. We're still looking at this CCG right here. And so the normal amino acid that should be there is glycine. But if we change this, now we're looking at, instead of the CCG, now we're changing a different nucleotide. So instead of mutating the third one, now we're mutating the first one. And so if we change that to a T instead of a C, that's going to change our mRNA, and then that's going to bring in the wrong amino acid. So instead of a glycine, we're going to end up with a serine instead. And so I think we talked about an example of, excuse me, sickle cell anemia. Did we? Yeah. Where one mutation in one gene, messes up the mRNA, and then that brings in the wrong amino acid. So in the normal red blood cells, you're supposed to have glycine, and it was replaced with a valine. Well, glycine is one of those negatively charged amino acids, and valine is extremely hydrophobic. So you can see the mismatch of amino acids. And so just that one mismatch is enough to make you have a disease. And so depending on the gene that it's in, if you're messing up your amino acid sequence, likelihood is you're messing up the way that it's folding and that all our groups are interacting with one another. All right, so we can also have a nonsense mutation. So in this example, we're looking at this piece of DNA, TTC, which gets made into AAG, which codes for lysine. But here, we're substituting an A instead of a T. And so when then you convert that into mRNA, now you've got UAG, which is one of those stop codons. So you now made a protein that only has a start, that only has a methionine. So I can guarantee you this protein is not going to be functional, because you need the whole length of the protein for it to fold and work properly. So if you accidentally make an early stop codon, that's going to cut your protein off early. So again, it's going to be non-functional. And then when it comes to the frame shift mutations, you can have different end results. So in this example, this isn't necessarily always going to be the case. It's just an example of what could happen as a result of a frame shift mutation. So again, here we have TAC, TTC, so forth and so on. But now we're going to insert an extra, we're just going to bring in another nucleotide out of the blue. And we're going to insert it in front of these two T's right here. So what that's going to do, this is a codon. Technically this is the codon. And this is a codon. But now, instead of having AUG, AAG, now we're having AUG, UAA. So it shifts the reading frame over by a nucleotide. And so now, all your amino acids, well, we're making an early stop codon, so there's not going to be any more amino acids. But in this case, this is a frame shift mutation leading to an early stop codon. Like I said, it's not always going to lead to an early stop codon, but it's a possibility. You could have a frame shift leading to a missense mutation. You could have a frame shift leading to all sorts of different things happening. But frame shifts are bad because, again, now that this codon has been messed up, that's going to mess up that codon and that codon and that codon. So normally when a frame shift mutation occurs, you're going to be completely messing up your protein and how it's made. Now this is looking at an insertion. Let's look at a deletion. So in this case, we're looking at AAA here, which gets transcribed, and UUU codes for phenylalanine. Well, now we're taking out one of those A's. So again, we're missing that U in the mRNA. So that's changing the reading frame. So instead of having phenylalanine and glycine, now you have leucine, alanine, and everything else after that point is going to be almost guaranteed to be the wrong amino acid. So this protein, because more than half of it has the wrong amino acids, is not going to be functional at all. And so you're always going to get a frame shift mutation if you have an insertion or a deletion in multiples of 1 or 2. But if